now to talk about this other tool, which is, I'm really excited to talk about it because it's a new tool. It's a tool that's only present in the GATK 2.0 or full version or the latest. It is a special tool that we developed for a very particular problem. We here at the BroadE quickly realized that we were quickly sequencing way more than we could process. And very quickly we faced the problem of how do we call this huge project with 12,000 BAMs? It's just too much data. We tried to run it with the regular pipeline, and the first thing that we got was just the file system blew up because there were too many files. The size of the BAM indices alone were too big to be loaded at the same time in memory. That's the very minimum we need to load just so we can scroll through the huge BAM files to get the pileups that we want to analyze. So what did we do? Well, back then when we faced this problem, we decided to chop the data into batches of 100 files, do the analysis independently on them, and then in the end use this very complicated statistical procedure to bring the results together and sort of give the project a combined result for the calling of all these samples. So this was the workaround that we used to do. Then came this tool. This tool provides a way for us to deal with large amounts of data by compressing the data. So we call it reduced reads. It's part of the data processing because we're still modifying the BAM file, but it only happens after all the processing that we've seen today. So after we have all the indels realigned, the reads are mapped, the indels are realigned, base quality scores are recalibrated. Now we have a pretty good final full BAM file. Now if you want to do a very large analysis, or if you want to just do things quickly, you can then reduce the file size by using reduce BAM. So that's why it's an addendum there, because it's a new tool and it comes in as the last step. And also because the BAM without the reduced reads is still analysis ready. It's just big and slow. So like I was saying, what is the purpose? So BAM files are huge, huge. A whole exome typically runs at about 10 gigabytes, and a whole genome runs usually at around 300 gigabytes. Think about 1,000 of those files. Think about 10,000 of those files. It's a lot of data. We cannot process that at the same time. And 12,000, as I said, was just the beginning. Ami just said he ran 24,000 BAMs, and we're just scaling out of proportions. We're thinking about doing 100,000 very soon. So we need to deal with the size of the file very quickly. Another big problem we have is the actual data is sequenced in many different centers usually, and we have to transfer these BAM files to one location so we can do the analysis. And transferring a few hundred terabytes is really, really, really difficult, let alone when you get to 100,000 BAMs, and we're talking about petabytes of data. It takes a long time. We have a lot of errors. You have to redo the transfers in many times. It's really not practical. The other problem is the time it takes to analyze things. Even if you have a huge machine that can handle all this data, it takes a long time to run even the simplest analysis just because of the volume of the data. And the complex to larger scale analysis is what I was trying to say about batching. The batching problems are really the result of combining the batches is really not ideal. It's not the full result. You lose a lot by doing that. And it would be good if we had a way to do this large-scale analysis without batching. So to achieve that, we created this tool. And just to give you an idea of what the benefits are, this is just running average time of running the unified genotyper, which is our SNP caller, on the original BAMs and the reduced BAMs. And those are just, I believe, 1,000 runs that we had. Just for testing, and the average time on the original BAM for this particular file was 40 minutes. On the same reduced file, it was about 1 minute, right? So this is how much faster it is just by getting rid of a lot of the data and instead placing it with information that will tell us all we need about the data. So what are the principles of this? The principle is basically throwing out data. The BEM file itself is very well compressed. So compression is not really just a matter of making things more efficient, packing things more efficiently. The BEM file does a good job about compressing the data. The problem is there's way too much data. When you look at the genome, usually large amounts of the genome is just boring and not informative. It's just a lot of reads that are telling you, yes, there is an A here, and it's exactly the same as the reference. Or there is a T here, it's not the same as the reference, but every one of the 200 reads that got through this site is telling me that this is a T. So all of these sections where the reads agree, we can basically skip it by just keeping enough information to tell us what was there. Was it 200 Ts? Great, that's all I need to know. I don't need to see 200 reads with a T. Just tell me that there were 200 Ts there. Somehow tell me what the quality of those Ts were, the mapping quality, and all the other parameters that we use for calling, and that's enough. Now, for the other reads, for the other parts where the reads don't really agree, we need to be a little more, a little smart about what we do with those regions. So the key concept of reduced reads is everywhere where the reads agrees, we will call it a consensus region, and we will compress as much as we can. We'll throw out all the data that we can throw out and replace that with meta information so that all the analysis tools can work with it. Now, for all the other regions, we'll call them variable regions, and those we're going to try to be smart about. We're going to try to keep as much information as we can so that all of the tools, the subsequent analysis tools, can be empowered to make the right decisions. So that's the whole philosophy. Throw away data that's not critical for making the right calls, and keep all the data that's necessary to disambiguate what's going on in the genome. So we call them consensus and variable regions. Variable regions are these windows around the disagreement. So if there is a disagreement of one site, then we'll draw a window around it, a window for just some padding on both sides, and keep all that data so that the subsequent analysis tools have more information than just that site to make the decision on what's going on there. The disagreement, as we're calling these variable regions, but this is a generic concept. These are just filters that you can plug into reduced reads and decide what makes a region a variable region. For example, the simplest example is a SNP. So if you're looking for SNPs only, then you can just say, well, a variable region is a region where in a site I have more than one nucleotide representing it. If there is more than one, do not touch that region. I need more information to decide what's going on there. So that's a very simple trigger. But you could have more complicated trigger. You could have coverage differences triggers saying that, oh, if at this region the coverage changes from X to Y and X is very, very different from Y, then trigger this as a variable region, or in other words, a region that we need to do more analysis to decide to disambiguate what's going on there. So these triggers are completely generic. We have, I think we ship it with SNP variants in general, SNPs, indels, long events. But you can do mate pairing, you can do coverage, you can do anything you want with the variant region triggers. In the end, after doing this, once we have a variant region, we also do a second step of downsampling. So what we do is, even though that region is complicated and we've decided not to touch it, not to compress it, we still don't really need thousands of reads to decide what's going on there. So we do apply a downsampling step that's very generous. We usually, I think the default is 250x, because with 250-fold coverage on any region, we're pretty sure we can determine what's going on there. So this is just a protection for problematic regions in the genomes, usually in the centromeres, where you have a lot of reads that got aligned there because it's very repetitive, and you can get very easily to 10,000 coverage, and you don't want to see all those 10,000. So that's just an additional step. And the latest feature here that I will just mention, it's not really that important, is that if you're doing tumor normal analysis, if you're working with cancer, and reducing may have, there are many caveats with reducing if you're talking about tumor normal. So to make those caveats less of a problem, we have this functionality of reducing samples together. So if you give it a tumor and a normal, they will be reduced together, and only regions, if any region is variant, either in the normal or the tumor, it'll be preserved on both. This way it'll give you the tools to distinguish what's going on either in the normal or the tumor by also looking at the data in the other sample. So this is just an optional tool. So this is what the compressed BEM looks like. At the top there, this is an IGV screenshot, just collapsed so that we can see hundreds of reads in a small screen. At the top, we have the original BEM with reads covering the whole, this is the whole genome sequencing, so you have reads everywhere. And down at the bottom, we have the reduced BEM, showing one single read representing the consensus of all the reads in the consensus region. And you have pile-ups, trimmed pile-ups around the variant region. Now, there's a few things I want to point out here. First, you see that the consensus read works for both sites, bases that match the reference and bases that are homozygous variants. So there are two homozygous variants there that mismatch the reference, but all the reads agree that that is a variant. It's a homozygous variant, and that's still a consensus to us. So we use the reference just for positioning, but we don't really use the reference for what bases should be there. We keep those bases as one read, and that's why we have the consensus read. In the consensus read, we also add information such as the average base quality score of all the reads that comprised this consensus. We also add a RMS of the mapping quality of all the reads that we use to make this consensus. We also keep the count, the number of reads that participated in this, and we also have a filter of reads that pass the quality requirements to be counted as one of the consensus reads. So in the end, this consensus read will give us information for each base of what is the depth, how many observations I had for that base, what is the average quality for each base, and for this consensus read, what is the general or average mapping quality of all the reads that formed this consensus read. So with all these information, we can actually make accurate calls just by looking at those values. Now for the variant regions, and they're pointed out here, this is a specific run where I set the option to trim the interval, trim the reads to the interval. You don't have to do that. It looks good. It looks like a perfect block because the reads that overlap the variant region were hard clipped. They were trimmed so that I only keep the information that I care about. For some analysis, this is not very good. You want to keep the whole read. You want to keep the information that goes beyond the variant region. And that's actually the default behavior of reduced reads. If you don't turn on the hard clip to interval options, it'll maintain the whole read here. So you would see something that looks more like a pile-up of reads here with the borders not matching, right? But only that region would be kept. And in the middle there, you'll see that there's the event. In this one, it's actually an insertion here that is shared by all the reads. And this is on purpose. We don't collapse insertions unless it's 100% in every read. Here you have, in these two, you have just a mismatch, a SNP, a heterozygous SNP, that will then be evaluated by the unified genotyper or the haplotype caller. All of these tools will be fully empowered by looking at the spiral up to make the right decision. What about a little bit more complex events, like a large indel or two events that are too close to each other? So in those cases, reduced reads will expand the variable region window to allow it to either combine the events or fully enclose the large event. So here we have a large indel, a 4-base variant indel. You see that the window just grows outside of it so that you still have that padding on both sides and include the whole event. And in that case, you have two events that are closer than the size of the window. So what happens is those two become one event, and the window goes around the two. So reduced reads will be smart about combining events that are very close together so that all of that data is preserved, and that is key information for the analysis tools, the subsequent analysis tools to distinguish whether or not this is a real event or an artifact. So basically, this is a very, very, very simple tool to run. You just need to take it, you only need your original BEM file, you run it, and you'll get a new compressed BEM file. The command line is as straightforward as it gets. You just need the reference, your original BEM file, and the output. There are a myriad of options that you can look at the website for clipping, for setting the thresholds, for setting triggers, turning them on and off, allowing soft clip bases and all these things. But we tend to believe that the defaults are pretty good. And, but we encourage you to go and check if you want to use any of the more advanced options. And the results look like I just showed before. So what are good diagnostic steps that you can do to make sure that everything worked? One thing that you can do is quickly do a SAMTools veal. It's just going to show you the reads, the actual reads in the BEM file. And you have to, after you run reduce reads, unless you've told the tool not to do so, it will strip out all the extra tags that are unrelated to the analysis that we do with the GATK. So there is an option to not allow it to throw away all the tags, but if you don't turn that on, it will throw away all the tags. So one quick check to see if the reads are reduced is to open the files and see if the tags are gone. Another thing to do is to look for consensus reads. You will see some reads that are 40,000 base pair long in your BEM file, right? If you have those reads, those are consensus reads, and their read names start with a C, and that's another good way to see if things actually happened. Now, good diagnostics are variant calling and coverage tests. So if you reduced your file, variant calling should yield the same results or very similar results. Of all our testing, we achieve very close to 100% sensitivity on the calls and specificity. So if you run variant calling on a small section of your BEM file, you should have very similar results to running the same unified genotype or haplotype caller in the original BEM. On the same note, you should get similar, very similar, not exactly the same results if you run a depth of coverage tool. Our current depth of coverage tool is called Diagnose Targets. You can look it up in the website. It's very simple to use too. Why do I say similar but not exactly the same? Because there are some caps. We have some caps on the coverage. So on a consensus read, if the coverage is beyond 128x, it'll be just capped at 128. Reason being, we could save more space by keeping the size of the number to a byte. And there is no difference practically if the read has no differences. We're talking about a consensus read, so everything had the same base. If you saw it 128 times, you were already extremely, extremely confident that that is the right call. So it's insignificant to say, oh no, it's not 128x, it's 2000x. It's overkill. So we decided to keep it short just to save some space. So if you run the diagnosed targets on those sections, you will see coverage being capped. That is also something that may be optional. For now, it's the way it's implemented, but we may change in the future. But for everything else, it should look exactly the same, bar the downsampling. I think this is, yeah, so this is where we are now with reduced reads, and I believe the next talks will be dealing with the variant discovery and genotyping. I am happy to take any questions. Yes?
What do you do with mate pairs?
What do I do with mate pairs? They're preserved in the variable regions, right? We don't do anything with them, we don't touch them, but for the consensus regions, they get lost. They're just erased. It makes no sense to talk about mate pairs. You still have the information on the variable regions of the start of your mate. If your mate got compressed into a consensus, you will still know where it would be, but the actual read got compressed and you don't have access to it anymore. So yeah, so you do lose mate information inside the consensus regions, where typically it wouldn't matter because those are consensus regions and you don't care where the mate went. But you still have some information that usually is enough inside the variable regions for you to look for the start location of your mate, the direction of your mate. You have that information still preserved in the read that passed through in the variable region. Yes?
Do you compress
I'm sorry, can't hear you.
You compress the heterozygous variant?
I compress yeah. So do I compress heterozygous variants? This is cutting edge new technology. We just developed that. Yes, we do. If you run the GATK 2.2 something, the latest one, it will do that. It's so brand new that I'm a little worried about talking about it because we may change things in the future. But yeah, we do have a new algorithm that will compress them if they're evidently het. If there's no doubt that there's only 2 alleles there, it's like 50-50. It's very clear it's a het. It'll compress it down to 4 reads, representing all the reads that are in the forward and reverse directions of each chromosome, each copy of the chromosome, right? So it reduces to 4. But again, this is brand new. That's why it's not in the presentation. Once we're satisfied and happy with it, it'll be available in the stable release. Yes?
How does duplicates, do you remove duplicates before you run this, or does this?
This is run after remove. So the question is, what happens to the duplicates? This runs after marking duplicates. One of the options is to get rid of the duplicates. That's one of the options. The default is to get rid of it. You can, however, say, no, I want to keep all the duplicates and include them in the analysis. We have never actually done that. We haven't found any particular analysis that this would be useful for, but the default behavior is just to get rid of the duplicates. Yes?
Do you have any comments about the UNBroadE's CRAM format? This is sort
Oh,
of revolutionary.
I have lots of comments. I can give you brief comments, and then we can talk offline if you want more about it. It's a great format. It's a good idea. It addresses a very different problem, the problem of storage, right? Although they do have some algorithms that try to be lossy, they're really not trying to address the problem of high volume calling and anything like that. We, with the reduced BAM, are not trying to solve the storage problem. We would not store reduced BAMs because they are very specific for analysis. You can't go back to it and, for example, realign. There's a new reference genome, realign, you can't do that. This is really just for processing. We call them analysis BAMs. They have a lot of good features, easy to transfer, easy to run analysis, very fast, and enables us to do large-scale analysis. But storage is still a problem. We want to store the original thing in case we want to redo the whole data processing pipeline. So for that, currently at the BroadE, we still store BAMs. But CRAM is this new format written by Iwan from the from EBI. He is compressing, he's doing a smart storage of the BAM file by inverting, but just basically flipping the way that the BAM file is recorded and doing it reference-based compression, which really yields really good results. It's approximately 4 times, without loss, approximately 4 times smaller than the BAM file, and without any loss. So, but still, it is the full file. So if you would run the CRAM file, it would take just as long as the BAM file, plus the time it takes to decompress and compress it. So it really isn't an analysis format. It's more of a storage format, which a lot of work is being done so that it's as efficient as the BAM file. So I would think of the CRAM as a great storage solution, perhaps the third version of the BAM file. Maybe it'll replace BAM file as the original file, but not as an analysis, as a quick, fast analysis tool like the reduced BAM file is. So those are 2 different niches. I have all the great things to say about the CRAM. They're doing a great job. We work closely with them in all of this, and I can talk a lot more about CRAM if you want, but it's important to make this distinction that CRAM is really a solution for storage and for the original BAM file, the size of the original BAM file. I believe maybe I blew through the slides, but the reduced BAM file is about 100 times smaller than the BAM file, whereas the CRAM is only 4 to 6 times. Those are the numbers that I saw last time I checked. So there's a huge difference there of size. Yes?
Is there an option to not compress the alternate homozygous reads?
The alternate homozygous reads. There isn't, but it's very easy to implement. So that could be something that we could add in the future. Not specifically the alternate homozygous, but all of the compression triggers have parameters that you can set. So you could, for example, say I only want to compress. You will define compression of homozygous to 90% of the reads or 75% of the reads. So you can be more lenient or more aggressive about your compression rate, but there's nothing about the opposite homozygous. It's very easy to implement. It could be something we could add, but it's not there. Do you have a question?
What happens to unmapped reads? Do they get excluded or?
They get removed. Yes, they get removed. Again, you can choose to keep them. It's an option, but by default, they get removed. That was the unmapped reads, in case you haven't heard. Yes? Tumor normal pairs?
Yeah. Do you have a more generic feature like that? So say, for example, you have no call in one of your samples and it's very region in another sample.
Okay. Yes, so the tumor normal is absolutely generic. We just presented it as tumor normal because that was the use case that we developed it for. But it's absolutely generic. If you run the tool with multiple samples, it'll act as if they were tumor normals. If you have 10 samples, if one of them has a variant region, all others will keep that region as variant. Okay, so it's absolutely generic. The terminology is just because that's how it came up. That's why we developed this mode. Okay? And then on that note, when you do that, you give n BEM files in, you get n BEM files out, right? They all come out reduced. So it doesn't compress it all to one BEM file. You get n BEM files out. Yes?
On the resulting VCF file that you run off of this BEM file and you're looking at a particular sample's metadata, if you're looking at a heterozygote, what would the resulting depth be for that genotype?
Okay, so the question is, when you make calls in this BEM, on a reduced BEM file, and you look at the metadata associated with the call, such as depth, what do you see? Hopefully, you will see exactly the same as you would see in the original file, barred things like downsampling, or if you're calling in a consensus region, you will see perhaps a compressed version of the actual depth if it's above 128. Bar those two details, you will see exactly the same because in the consensus region, we keep the number of bases that passed all the filters that were used for that compression. So if you had 38 A's, that number 38 will be used by the unified genotype or the haplotype caller to come up with the decision of the genotype. And in the variant region, you still have all of the reads there. So it's basically the same thing as running in the original BEM. So most of the metadata looks the same. We have done extensive analysis on all of our metrics that we use for calling. So things like haplotype score, mapping quality score, mapping quality, base quality by depth, all the values that we use for filtering evaluation. This is going to come up tomorrow on the variant quality score recalibration talk. We have done extensive analysis to make sure that reducing the BEM wouldn't impact the filters and all of the annotations that we make on those metadata. And it really worked very well for everything that we've done so far. All right. How are we doing on time? We're good. All right, thank you.